regulation, microRNA. For a long time, however, microRNA was believed to be an oddity peculiar to C. elegans. This perception changed 7 years later when Gary Ruvkun, in the year 2000, identified a second microRNA produced by the LET7 gene in the world. Unlike LIN4, LET7 was found to be highly conserved and is present in human and most of the animal kingdom. This finding made the previously dormant microRNA explode. As it turned out, many microRNAs are highly conserved throughout evolution over several 100 million years. In more complex organisms, a larger number of microRNAs have evolved, with humans having more than 1,000 distinct microRNA genes. Today, we understand that the majority of all genes are regulated by microRNAs. Every microRNA regulates several genes, several mRNAs, and each mRNA is often regulated by many distinct microRNAs, creating a robust system for gene regulation. The seminal discovery of microRNA has introduced a new and unexpected mechanism of gene regulation. MicroRNAs are important for our understanding of embryological development, normal cell physiology, and diseases such as cancer. As an example, tumors often perturb microRNA networks in order to grow. Victor Ambros and Gary Ruvkun have jointly been awarded this year's Nobel Prize in Physiology and Medicine for their discovery of microRNA and its role in post-transcriptional regulation. I now hand back to Professor Thomas Perlman, the Secretary General of the Nobel Assembly.
Thank you very much, Ulla. So with this, we would be happy to answer questions. And you've already been introduced to Professor Kempe, but on my side, I also have the chair of the Nobel Committee, Professor Gunilla Carlsson-Hedestam, and member of the Nobel Committee, Professor Richard Sandberg. We have a question up there first.
Okay, thank you. I'm Anna-Lena Arn from the TV4. Do I need a microphone? No, maybe not.
Yes, please.
I have a question regarding your second last slide, if you could get back to that one. I wonder how it's possible for the microRNA to get to the mRNA first, so to speak, so that it shuts down this mRNA and the protein is not made. I mean, if the mRNA is made first, then the protein would be made anyhow. And I also wonder what the different cell types, why is the microRNA, a special microRNA, active in one sort of a cell and not in another sort of a cell?
Yes, these are quite advanced scientific questions, and I'll turn to Professor Richard Sandberg.
So regarding the first question, so we skipped a lot of detail in this presentation, but the microRNA strand is part of a protein complex that finds the mRNA in the cytoplasm. And there it can recruit other proteins that help shorten the poly-A tail, which leads to either translational inhibition or mRNA degradation. The second question about microRNA genes, I mean, they are also transcribed, so they're copied from the DNA into their RNA form, they're processed, and that can be done cell type specifically. So a specific microRNA will end up in a muscle cell, whereas other microRNAs end up in, for example, nerve cells. And because of that, they can have cell type specific effects on the mRNA.
Hey, my name is Steffen Trump from the German news agency dpa. Thomas, it's early morning in the US, so my question would be, have you already reached one of the laureates? And yeah, what did they say for being awarded?
Yeah, nowadays it tends to be a little more difficult. People have mobile phones on silent, but I was able to wake up Gary Ruvkun. So I think he had a landline. I was able to reach him. His wife answered, and it took a long time before he came to the phone and sounded very tired. But he quite rapidly was quite excited and happy when he understood what it was all about. He was so enthusiastic, and his wife too, so he handed over the phone to his wife, who also wanted to talk to me for quite a while, and they were thrilled about the prize and coming to Stockholm in December. I was not able to reach Victor Ambros yet. I left a message on his mobile phone and hope he gives me a call soon.
Hello, my name is Bogun Radeski. I'm from the Polish television. A question I have is a little bit of comparison to the last year's Nobel Prize, because last year with the Nobel Prize for the mRNA, the thing that we are most excited about, obviously, is what it can be used for. Not only COVID vaccines, but also the prospect of cancer vaccines and solution to many diseases that we're facing today. So what about microRNA? How does it compare, and what could be the application of the discovery by this year's laureates? Thank you.
I think this is a very good question for Professor Karlsson-Hedestam.
Yes, thank you for that. So this year's prize is definitely a physiology prize. It helps us, our basic understanding of all of the things you heard about, how cells differentiate and become specialized. Last year, of course, a much more applied discovery that was translated into vaccine development. So two quite different prizes. But that said, having a basic understanding is, of course, the first step towards developing applications. So although there are no very clear applications available yet using microRNAs, understanding them, knowing that they exist, understanding their kind of regulatory networks is always the first step. So this could still take a while. There are quite a lot of trials ongoing, and not only against cancer, but also in other diseases, the cardiovascular, kidney diseases. There are other trials ongoing, but nothing that is very sort of close to an actual sort of application. So, but we want to emphasize that the importance of understanding the basic functions, and that is always the first step towards using this knowledge.
Det kommer en.
I wonder if you are aware if the laureates have any patents on their discovery, and also if you could describe a little bit more about their collaboration. They were postdocs together, and how are their friends hanging out in the lab, or what's their collaboration like?
Olle Kempe, maybe you want to...
Well, they were postdoctoral fellows together, but then ran separate labs and focusing partly on different aspects of the microRNA. So the big findings were from 1993, when the first microRNA was discovered by Victor Ambros, the Lin4 mutant. And then 7 years later, in 2000, Ruvkin came with the second. So if there had been any patents, they're no longer valid.
Right. Yes, up here.
Hi, this is Sunlight Life Weekly from China. A following up question about the clinical application, as Professor Haddison mentioned that because microRNA has been widely used as a research tool to modulate gene regulation, what is the main challenges to develop a microRNA as a drug or for its clinical applications?
Olle Kempe, maybe you can.
Well, I think there are many problems. I mean, one thing is to deliver it to the right tissue. The second problem is, of course, that microRNAs bind several targets. So you very easily get off-target effects if you directly bind to one microRNA. But there might be ways around that. Tumors quite often perturb the microRNA networks, and they can do it by deleting the genes or by mutating genes that process the microRNA, or they can increase the production of RNA binding protein that binds the microRNA. And in that case, there are promising first tests to see if you can modulate the RNA binding proteins. But to deliver microRNAs to cells and think that you get one effect, I think will be very difficult.
Yes. Här vid kameran.
Thank you. Hello, Paul Rees, Al Jazeera English. If you had to explain in just a few words, as some of us may have to do later, what the difference is between mRNA and microRNA, so that perhaps people don't get confused between this and last year's.
Yeah, I'll ask Rickard Sandberg to explain the difference.
Yes, so I mean mRNA is the very fundamental intermediate between our DNA and the proteins, and it's sort of the step that carries the information from the nucleus to the protein-producing machinery. MicroRNAs, on the other hand, they are very short, and their primary role is to act as itself, as its RNA form, where it instead interacts with the mRNAs to tune or quite dramatically affect what amount of proteins can be made from it, or should we degrade the mRNA.
So nothing to do with COVID vaccines this time?
Sorry?
So nothing to do with COVID vaccines this time?
No, not really.
Let's see, is there any other question? I know it's, as is usual, that many are eager for the interviews instead after the press conference, but still, if there is any pressing question, there is still time. If anyone would be interested in something. If not, then thank you so much for coming here today, and we'll see several of you at interviews. Thank you.
Ja. Jag har blivit ombedd av Ann-Marie att lotsa dig bort och